Welcome to the deep dive. Today, we are really jumping into something critical, the science of microbial control. This is the kind of foundational knowledge that, well, it underpins so much modern medicine, public safety, even the food you eat.
It really does.
We're taking a close look at the physical and chemical tools, the weapons, if you like, used in this constant battle against microscopic things trying to grow where we don't want them.
And it's crucial for you, the listener, to grasp not just the words, but the precise way these methods are used. We're not, trying to wipe out every microbe on Earth. That's impossible and, frankly, undesirable.
Right.
Our goal is much more specific. Getting rid of or stopping the growth of the particular microbes that cause infections, disease, or make our food go bad. It's about reducing their numbers down to safe levels, verifiably safe levels.
Okay, let's break this down then. Our source material today really lays out these methods, giving us that practical know-how needed for, well, anyone in healthcare or related fields. And when you think about the stakes, the history is actually kind of fascinating. People have been trying this forever, haven't they? Like the ancient Egyptians using spices for mummification, or Alexander the Great telling his soldiers to boil their water. Basic stuff, but it's a start.
Absolutely. But the real game changer, especially for high-stakes situations like hospitals, that really kicked off more recently. You think of Ignaz Semmelweis back in the 1840s.
Oh yeah, the hand-washing guy?
Exactly. He slashed infection rates after childbirth just by making doctors wash their hands with chlorinated disinfectants. Simple, but revolutionary. And then Joseph Lister built on that, pioneering aseptic surgery using carbolic acid.
So it wasn't just about being clean, it was active, targeted microbial control.
Precisely. It established this absolute need for it in any professional setting where health is on the line.
Okay, before we get any deeper, we absolutely have to nail down the vocabulary. Because messing up terms like sterilization and disinfection, well, that can have serious consequences.
You are absolutely right. Precision here is paramount. We can sort of group the key terms by what they achieve, their outcome. At the very top, the most extreme is sterilization.
Right.
Total annihilation.
Pretty much. Complete destruction and removal of all microbial life. And that includes the really tough forms like bacterial endospores and even prions. Nothing survives.
And then there's that related term, commercial sterilization. Sounds similar, but it's different, isn't it?
It is, and it's a perfect example of applying the science practically. Commercial sterilization is specifically for the food industry. It uses enough heat treatment just to kill the endospores of Clostridium botulinum.
The botulism one.
The very one. You get rid of that deadly toxin risk, but you don't necessarily achieve absolute 100% sterility of everything else. It's a targeted approach, a necessary trade-off sometimes to keep the food, well, edible.
Okay. Moving down the scale a bit, how do we separate actions on, say, a counter versus on skin?
Good distinction. We use the term disinfection when we're destroying vegetative pathogens. Those are the actively growing ones on inert, non-living surfaces, like a countertop, a bed rail, medical instruments.
And if you do that same thing on living tissue, then it's called antisepsis. The process is antisepsis, and the chemical you use is called an antiseptic. Think iodine on a cut or alcohol wipes.
Got it. And then we have the terms that mean just reducing the numbers.
Exactly. Sanitization is about lowering microbial counts to safe public health levels, usually on things like eating utensils or maybe food prep surfaces. It implies cleaning to a safe standard, but not necessarily disinfection.
Okay.
And finally, degermination. This one's purely mechanical removal. Think about swabbing the skin with that alcohol pad before injection.
Ah, right. You're physically lifting the microbes off.
Precisely. You're not necessarily killing them all right there, but you're removing them from that specific limited area.
Okay, so we've applied one of these methods. What does microbial death actually mean scientifically? How do we know it worked?
It means the permanent loss of the microbe's ability to reproduce. That's the key. Even if you put that treated organism back into the most perfect nutrient-rich environment for it, it just can't replicate.
It's done for.
It's done for. The treatment has caused irreversible damage, usually to its essential structures like proteins or its DNA.
Now, this is where it gets mathematically interesting for me. Microbial death isn't like flipping a switch, is it? It's exponential. Which leads us to the decimal reduction time, or DRT.
Yes, the DRT. It's such a fundamental concept. It's the time, usually measured in minutes, that it takes to kill 90% of a specific population of bacteria at a specific constant temperature.
90%. Why is that specific number so important?
Because it shows the exponential nature. If you start with, say, 1,000,000 bacteria, killing 90% leaves you 100,000. Kill another 90% of those, you're down to 10,000. then 1,000, then 100, then 10.
Ah, I see. So the time it takes depends hugely on how many microbes you started with.
Exactly. This DRT calculation directly tells you how long you need to apply the heat or the chemical to reach a safe level, or even sterility. The initial bioburden, the starting number of microbes, is critical.
And even before picking a method, you need to survey the scene, right?
Oh, absolutely. How well any method works depends heavily on several things. What are you treating? Is it something delicate that heat would destroy? What are the environmental conditions?
Like, is there gunk in the way?
Precisely. Is there a biofilm? Biofilms are notoriously hard to penetrate with chemicals. Is there organic matter present, like blood, pus, or feces? Because that stuff can actually inactivate some disinfectants.
Mm-hmm.
And maybe the biggest factor, how susceptible is the microbe you're actually trying to kill? They aren't all created equal.
Right. Let's talk about that hierarchy of resistance. It's quite a spectrum, from the easy targets to the almost indestructible ones.
It really is. Down at the more fragile end, you've got things like enveloped viruses, think influenza or HIV, and most gram-positive bacteria. They're generally the easiest to eliminate.
Okay.
Then you move up the ladder, you get protozoan cysts, non-enveloped or naked viruses, which are tougher, fungi, and then things like mycobacteria, the ones that cause tuberculosis. They've got that waxy cell wall offering extra protection.
But the real heavyweights, the ones that cause major headaches in hospitals and food safety, are the bacterial endospores and prions.
Definitely. Bacterial endospores. These are formed by bacteria like Clostridium, remember botulism,
Mm-hmm.
and Bacillus. They aren't reproductive structures. They're dormant survival pods.
Built like tanks.
Essentially, yes. They can survive boiling, most common disinfectants, even radiation doses that would kill other things. If your method doesn't reliably kill endospores, you absolutely cannot call it sterilization, period.
And it's worth maybe quickly contrasting those with protozoan cysts. Cysts are tough, but endospores are tougher.
Much tougher, especially chemically and thermally. Cysts are resistant. Yes, they help protozoa survive harsh conditions, but bacterial endospores take that resistance, especially to heat, to a whole different level.
Okay, and then at the absolute peak of this resistance mountain.
Prions. These are misfolded proteins, not even cells, that cause fatal neurodegenerative diseases like Creutzfeldt-Jakob disease or mad cow disease.
Just protein.
Just proteins, but incredibly stable and resistant. They are the hardest things known to inactivate. Getting rid of them often requires really extreme measures, combining chemical and physical methods, like autoclaving contaminated surgical tools in a strong sodium hydroxide solution. They are the ultimate sterilization challenge.
Wow. Okay, so we've identified the enemy, ranging from weak to terrifyingly strong. Now let's deploy the arsenal, starting with physical methods, and heat seems like the cornerstone.
It absolutely is. Heat works fundamentally by messing up the cell's machinery. It denatures proteins, basically unfolds them so they can't function, and inactivates essential enzymes. Game over for the microbe.
And we split heat into dry heat and moist heat.
Right, dry heat, like in a hot air oven, needs higher temperatures, maybe 160 to 170 degrees Celsius, and much longer times. We're talking hours to achieve sterilization. Think baking things sterile, or even incineration, just burning things to ash.
But moist heat, that's the real powerhouse, isn't it? Works faster at lower temps.
Much more effective. Why? Because water is way better at transferring heat energy into the microbial cell than dry air is. It penetrates faster and denatures those proteins more efficiently.
And we have specific terms to measure that effectiveness.
We do. The thermal death point, TDP, is the lowest temperature needed to kill all microbes in a standard liquid suspension in exactly 10 minutes. And the thermal death time, TDT, is the minimum time needed to kill all microbes in that liquid at a given constant temperature. They help standardize things.
Okay. And the undisputed king of moist heat sterilization is the autoclave.
The autoclave, yes. It's essentially a pressure cooker, uses steam under pressure. By cranking up the pressure inside to about 15 pounds per square inch, PSI, above atmospheric pressure, the boiling point of water jumps from 100 C up to 121 degrees Celsius.
And that combo is lethal.
Extremely lethal. That steam at 121 C under pressure penetrates materials quickly and kills everything, including those tough endospores, usually in about 15 to 20 minutes depending on the load. It's the gold standard for sterilizing most lab equipment, surgical instruments, dressings, culture media, anything that can withstand the heat and moisture.
So if the autoclave is so good, why would we ever use anything else heat-wise? Is it just because some things can't handle 121C?
That's the main reason, yes. Some materials, like certain plastics or delicate culture media, would be completely destroyed by autoclaving. For those, sometimes you might use tyndallization.
Tyndallization sounds old school.
It is a bit, and less common now, but it's clever. It's intermittent sterilization. You steam the material usually at 100 C, just boiling for maybe 30 minutes on day one. That kills most vegetative cells.
But not spores.
Not reliably. Then you incubate it overnight. The idea is that any surviving spores will germinate in the nice warm media into new heat-sensitive vegetative cells.
Ah, sneak.
Then, day 2, you steam it again, killing those newly germinated cells. Repeat incubation, and steam again on day 3 just to be sure. It's slower, requires care, but it can sterilize heat-sensitive liquids without high temperatures.
Okay. What about going cold? Refrigeration, freezing, we use those all the time for food.
Right, but it's important to remember that cold temperatures are generally bacteriostatic, not bactericidal.
Meaning they stop growth but don't kill.
Exactly. They slow down or pause microbial metabolism and reproduction, which is great for preservation, but most bacteria will survive refrigeration or freezing and start growing again once warmed up. Freezing can kill some due to ice crystal damage, but it's not reliable for sterilization.
But there's a special kind of freezing for preservation?
Yes, lyophilization or freeze drying. This is really cool. You rapidly freeze a biological sample like a bacterial culture you want to save, and then you put it under a strong vacuum. This causes the frozen water, the ice, to sublime, meaning it turns directly into gas without melting first.
Skips the liquid phase.
Right, and that avoids forming large ice crystals that would damage the cells. You end up with the dry powder that can preserve microbes or biologicals for years. Just add sterile water, and they often come back to life.
Okay, beyond temperature, radiation. Used a lot for single-use medical stuff, right?
Absolutely. We mainly use 2 types. First is ionizing radiation. This includes gamma rays, from cobalt-60 usually, X-rays, and high-energy electron beams. They're powerful.
How do they work?
They have enough energy to knock electrons off atoms and molecules as they pass through. This creates ions and highly reactive free radicals, like hydroxide radicals, which just wreak havoc on cellular components, especially DNA. They shred it, basically.
And that's good for Napper.
Sterilizing things that can't handle heat, particularly prepackaged disposables like plastic syringes, gloves, sutures, even some pharmaceuticals. Gamma rays and electron beams are common commercially. X-rays penetrate the best, but generating them takes longer and is more complex.
And the other type, the UV lights we sometimes see in labs or HVAC systems.
That's non-ionizing radiation, specifically ultraviolet UV light, usually around 260 nanometers wavelength. It's not as energetic as ionizing radiation.
So how does it kill microbes?
Its main trick is causing specific damage to DNA. It causes adjacent thymine bases or cytosine in the DNA strand to covalently bond together, forming dimers. Think of it like welding 2 adjacent rungs of the DNA ladder together.
Jams up the works.
Exactly. It messes up DNA replication and transcription. But UV has a huge limitation: penetration. It doesn't go through materials well at all. Not solids, not glass, not even very far into liquids unless they're clear and shallow.
So it's mainly for surfaces and air?
Primarily, yes. Disinfecting exposed surfaces in operating rooms or labs, disinfecting air in ducts, or treating water if it's flowing in a thin film. Germicidal lamps.
Okay. Last physical method, filtration. If you can't heat it and UV can't reach it, you filter it.
Precisely. Filtration is simply mechanical removal. You pass a liquid or gas through a filter membrane with pores so small that microbes can't get through.
What kind of things do we sterilize this way?
It's essential for heat-sensitive liquids. Think certain antibiotics, vaccines, serum solutions used in cell culture, some vitamins, things that would be denatured or destroyed by heat. You just physically sieve the bacteria out.
What size pores are we talking?
For an moving bacteria, the standard membrane filter pore size is typically 0.22 micrometers, sometimes 0.45 micrometers. Viruses are much smaller, so they'd pass through these filters unless specialized ultrafilters are used.
Right. Okay, that covers the physical attacks. Now let's move to the chemical arsenal. Disinfectants, antiseptics. Lots of different groups here.
It's a huge area, yes. We really need to focus on the major classes and how they work, their mechanism of action. Let's start with phenols and phenolics. Phenol itself isn't used much anymore, too irritating, but its derivatives, phenolics, are common.
Like in Lysol.
Exactly. Things like O-phenylphenol are active ingredients in Lysol.
Okay.
They work primarily by disrupting cell membranes, making them leaky, and denaturing proteins. A big advantage is they remain active even if there's organic matter around, like pus or feces, which can neutralize other chemicals.
Good point. Then we have the halogens, chlorine and iodine, household staples.
They are. Chlorine, often used as sodium hypochlorite, that's bleach, or calcium hypochlorite, or chlorine gas for water treatment, is a powerful oxidizing agent. It just oxidizes and destroys cellular components, enzymes, pretty much everything. Iodine is similar, often used as a tincture in alcohol, or as an ionophore like betadine, where it's combined with an organic molecule for slower release and less irritation. Great antiseptic.
Okay, now let's hit that crucial detail about alcohols, ethanol, isopropanol. They denature proteins, dissolve lipids, but why is 70% alcohol the magic number, better than, say, 95%?
Ah, yes, this is such a key concept, and it seems counterintuitive. You'd think more alcohol is better.
Yeah, pure alcohol should be strongest.
But protein denaturation, the main way alcohol kills, actually requires water to happen effectively. If you use 95% or absolute alcohol, it causes rapid coagulation of proteins right on the surface of the microbes.
Like cooking an egg white instantly.
Kind of, yeah. It forms this hardened protein shell almost immediately, and that shell actually prevents the alcohol from penetrating deeper into the cell to kill it properly.
Mm-hmm.
Whereas a 70% solution, roughly 60–80% is the effective range, has enough water mixed in. The denaturation happens a bit slower, allowing the alcohol to fully penetrate the cell before the proteins coagulate, leading to a much more effective kill. Water is essential for the process.
That is a fantastic takeaway. Okay, what about quaternary ammonium compounds, the quats? We see those in surface cleaners, disinfectant wipes.
Yep, quats are very common. They're basically cationic detergents. Think of them like soaps, but with a positive charge. That charge helps them disrupt cell membranes. They're good bactericidal agents, especially against gram-positive bacteria, and they kill enveloped viruses. Plus, they're stable, low toxicity, non-corrosive.
Sounds great. Any downsides?
Their main weakness is their spectrum. They're generally considered low-level disinfectants. They don't reliably kill non-enveloped viruses, mycobacteria, or endospores. And things like soap or organic debris can inactivate them. So, good for general sanitation, but not for high-level disinfection or sterilization.
Okay. For the really heavy-duty chemical jobs, maybe even chemical sterilization, we need the alkylating agents.
Now, you're talking serious chemicals. These are highly reactive compounds that work by adding alkyl groups, like NiCH2CH3, to important molecules in the cell, especially proteins and nucleic acids, DNA and RNA. This cross-linking and inactivation is lethal.
Examples.
Two big ones. Glutaraldehyde. So liquid, often uses a 2% solution. It's less irritating than formaldehyde, but still very effective. It can achieve high-level disinfection in minutes, but for true chemical sterilization killing spores, you need long immersion times, like 10 hours or more. Used for sterilizing heat-sensitive medical equipment like endoscopes.
And the other big one is a gas, right?
Right, ethylene oxide, ETO. This is a highly penetrating gas used for sterilizing massive amounts of prepackaged medical devices, plastics, instruments, anything that can't take heat or moisture. Think catheters, heart valves, syringes, loaded into a special chamber.
Sounds powerful, but maybe dangerous.
Extremely. ETO is highly flammable, explosive, and it's a known carcinogen. It requires very specialized equipment, long aeration times after sterilization to remove residual gas, and strict safety protocols. It's critical for healthcare, but definitely not something used casually.
Understood. So we have all these chemicals making claims. How do we actually test if they work as advertised?
There are standardized methods, mainly regulated by agencies like the EPA or FDA. The classic one is the use-dilution test.
How's that work?
You take small stainless steel cylinders, dip them in a standardized broth culture of specific test bacteria like Staphylococcus aureus, Pseudomonas aeruginosa, Salmonella. Let them dry briefly.
Oh,
So now they're contaminated. Then you immerse these contaminated cylinders into the disinfectant solution being tested at the recommended dilution for a specific time, usually 10 minutes. After that, you take them out, rinse them briefly, and pop them into tubes of sterile growth medium. Incubate.
and if the disinfectant worked?
The broth stays clear, no growth. If the disinfectant failed to kill the bacteria on the cylinder, the broth will turn cloudy, turgid. That indicates growth. You usually test multiple cylinders to ensure reliability.
Makes sense. What's the other common visual test with the Piper discs?
The disk-diffusion method. This one's simpler, often used for screening or comparing antiseptics maybe. You spread your test bacteria evenly over the surface of an agar plate to create a lawn of potential growth.
Right.
Then you take small filter paper discs, saturate each one with a different chemical agent you want to test, and place them carefully onto the agar surface. Then you incubate the plate.
And you look for clear zones.
Exactly. If the chemical is effective, it will diffuse out from the disc into the agar and inhibit or kill the bacteria nearby. This creates a clear circle around the disc where nothing grew. That clear area is called the zone of inhibition. The bigger the zone, generally, the more effective the chemical is against that specific microbe.
Got it. Okay, let's pivot and apply all this directly to something we encounter daily, the food industry. The goal here isn't just making food last longer, it's preventing foodborne illness, right? Millions get sick every year.
Absolutely. Preventing spoilage is important economically, but preventing pathogens like Salmonella, E. coli, Listeria, and especially Clostridium botulinum from causing disease is the critical public health mission.
And the cornerstone method here seems to be pasteurization, thanks to Louis Pasteur.
Indeed. Back in 1857, he figured out that applying mild heat to liquids like wine and later milk could kill off most of the spoilage microbes and potential pathogens without drastically changing the taste, texture, or nutritional value. It was revolutionary for food safety and shelf life.
But crucial point, pasteurization is not sterilization, is it?
Absolutely not. That's a key distinction. Pasteurization achieves a significant reduction in microbial numbers, a log reduction, killing most vegetative pathogens and spoilage organisms, but heat-resistant, non-pathogenic bacteria, Thermodurix, can definitely survive. That's why pasteurized milk still needs refrigeration and eventually spoils.
And the methods have gotten faster since Pasteur's time.
Oh yes. The classic batch method, like 63 degrees C for 30 minutes, is less common now for milk. We mostly use HTST, which stands for high temperature, short time. It's typically heating milk to 72 degrees C for just 15 seconds, followed by rapid cooling. It's efficient for large volumes.
And then there's this stuff that sits on the shelf for months.
That's usually treated with UHT, ultra-high temperature processing. This involves heating the liquid to much higher temperatures, maybe 138 to 140 degrees Celsius, but for only a tiny fraction of a second, like 1 to 3 seconds. Then it's aseptically packaged into sterile containers.
And that does approach sterility.
It kills pretty much everything, including spores, leading to a product that's shelf-stable for months without refrigeration until you open the package. Think those little cartons of milk or cream.
Okay, now the big warning you always hear about home canning. Low-acid foods like vegetables, meats. Why must they be pressure canned? Why isn't just boiling them in a water bath enough?
This comes right back to our old friend Clostridium botulinum and its endospores.
You hit.
Low-acid foods, pH above 4.6, don't inhibit the growth of C. botulinum the way high-acid foods like fruits or pickles do. Boiling water only reaches 100 degrees C to 112 degrees F at sea level. That temperature is not sufficient to reliably destroy C. botulinum endospores.
They can survive boiling.
They can.
Yeah.
And if they survive in that anaerobic low-acid canned food environment, they can germinate, grow, and produce the deadly botulinum neurotoxin. Pressure canning allows the temperature inside the canner to get much higher, typically 116 degrees C, 240 degrees F, or more because of the steam pressure. That temperature will destroy the spores, making the food safe. It's absolutely critical for low-acid foods.
Non-negotiable. Okay, tying back to our earlier discussion on radiation, what about food irradiation? How does that fit in?
Food irradiation uses the same ionizing radiation, usually gamma rays or electron beams that we talked about, for sterilizing medical supplies. It's used to control spoilage microbes and kill pathogens like Salmonella or E. coli in foods like meat, poultry, spices, and fresh produce.
Does it make the food radioactive? That's a common concern.
Absolutely not. The radiation passes through the food, like light passes through glass. It kills the microbes by damaging their DNA, but it doesn't leave any radioactive residue behind. It's endorsed by major health organizations worldwide as safe and effective.
Does it kill everything?
It's very effective against bacteria and parasites. Viruses and prions, however, because they have such small or non-existent nucleic acid targets, are generally much more resistant to the radiation doses approved for food. So it's not a silver bullet for all potential contaminants, but it's a valuable tool.
And finally, lots of traditional food preservation tricks basically use the same principles we've been discussing, just in simpler ways.
Exactly. Drying or desiccation removes water, inhibiting microbial metabolism. Pickling uses acid, low pH, which many bacteria can't tolerate. Curing meats uses high concentrations of salt and sometimes nitrites, which inhibit growth through osmotic pressure and specific enzyme inhibition.
And jams and jellies.
Same principle as curing, but with sugar. The incredibly high sugar concentration creates massive osmotic pressure, drawing water out of any microbial cells and stopping their growth. It's all about creating an environment hostile to microbial life.
So wrapping this all up, what are the absolute must-remember takeaways from this deep dive into microbial control?
Okay, number one, know the precise definitions. Sterilization means complete kill, including spores. Disinfection kills vegetative pathogens on surfaces. Antisepsis does it on living tissue. Sanitization just reduces numbers. Get those straight.
Right. And number two?
Endospores and prions are the supervillains, exceptionally hard to kill. Most methods short of sterilization won't touch them reliably, especially see botulinum spores in food and prions in healthcare settings.
And number three, maybe that alcohol detail.
Definitely. Remember why 70% alcohol works better than 95% water is required for the protein denaturation process to penetrate and kill effectively. Don't forget that one.
Excellent. So looking ahead, what are the ongoing challenges or maybe future directions in this field?
Well, the battle definitely isn't over. Biofilms, those slimy layers microbes form on surfaces, remain a huge challenge because they're so hard for chemicals to penetrate. Finding better ways to tackle biofilms is a big research area.
And resistant strains.
Always, we're constantly facing new or evolving drug-resistant organisms like MRSA, methicillin-resistant Staphylococcus aureus, or CRE, carbapenem-resistant Enterobacteriaceae. We need continuous development of new antimicrobial agents and strategies, maybe even things like phage therapy, to stay ahead. And finding less toxic, more environmentally friendly options is also key.
And there's that interesting question raised in the source material too, the can you be too clean idea, the hygiene hypothesis.
Right. It's that fascinating balance. We absolutely need rigorous microbial control in healthcare and food safety to prevent disease. But some research suggests that maybe excessive cleanliness, especially early in life, might limit our immune system's exposure to diverse microbes needed for proper development, potentially contributing to allergies or autoimmune issues later.
So protect from pathogens, but maybe don't sterilize our whole lives.
It's a complex area. Finding that right balance between necessary sanitation for preventing acute infections and allowing enough microbial exposure for healthy immune system training, that's a really important ongoing discussion in public health.
Food for thought, definitely. Well, thank you so much for walking us through all of that, and thank you for joining us on this deep dive. You should now have a much clearer picture of the essential science behind controlling the microbial world around us, crucial knowledge for anyone heading into healthcare or related fields.